Thank you very much for the invitation to speak, and thank you, Veronica, for the glowing. You have, yes. And I apologise for my voice. It might at one point lapse into sort of pure Barry White, because I have a bit of a cold and sore throat. So I apologise if I do. So I was asked to cover two quite different topics in a way, but I came up with a title that sort of linked them. The second part, which will be about the regulation of human genome editing, will have overlapped a little bit with Richard's, so maybe I can cut that a little bit shorter. But okay, so the first part is the regulation of SOX9 gene expression during sex determination, tipping the balance. So, of course, genes have two components to them. They have the protein coding part, and then they have the part that's involved in regulating whether they're active or not. And that is very important in many biological contexts, and as you'll see, hopefully, it's important in the context of whether we develop as males or females. So I've worked on sex determination, as Veronica said, for many years. So sex determination is the decision to develop as male or female, and how that switch actually operates. We can sort of distinguish a little bit from sex differentiation, which is how that decision is then translated to all the differences between the sexes in terms of anatomy, physiology, behavior. So many reasons for doing research on this, ranging from pure interest. There have been theories about how you get boys and girls for thousands of years. One of my favorite theory was one of the ancient Greek philosophers, Anaxagoras, proposed that semen from the left testis gave rise to girls and from the right testis to boys. That's a theory that can be tested very easily. It's obviously socially important, whether you have a boy or a girl is one of the first questions asked of newborn parents. It's something that we supposedly have to put on all sorts of different forms. I'm not sure I always agree, but we're supposed to do that. But the reason I study it is because it's a paradigm of how decisions of cell fate occur in embryonic development. Also because the gonads are quite relatively simple organs, it's a paradigm of how organs are formed, how they develop. It's clinically important. I will not have time to go much into this, but you'll see from the talk that disorders of sex differentiation, such as you can have complete sex reversal, where the individual develops the wrong phenotypic sex compared with their chromosomal sex, or more troubling is where you have a mixture of male and female characteristics, so intersex. Obviously, often it's associated with infertility. You've got other issues like gender dysphoria. You have a whole variety of conditions and mechanisms, so diseases and mechanisms that exhibit a distinct sex bias. Some mechanisms are very different between males and females. At least in mice, you can make mutations in specific genes which severely affect cognitive ability in male mice, but have no effect on females at all, for example. So knowledge of genes and processes involved allows diagnosis, counseling, appropriate choice of treatment, and development, perhaps, of novel clinical options. So there are many different mechanisms of sex determination have evolved. So these fall largely into two classes. There's environmental mechanisms where essentially all individuals within the same species have the same genetic makeup, roughly. But it's actually the environment which influences whether which sex develops. So you see this in many species of fish and many species of reptiles, not all, but many. So, for example, it's the temperature at which the embryo is incubated during a critical period of its development that determines whether you're going to get male or female crocodiles or turtles. You can, in some species of fish, you can have, for example, a school of fish. They're all female except for one male. If the male dies or swims away, then the dominant female, largest female, simply notices there's no male there, and she then turns into a male. Doesn't happen in humans. It would be sort of socially interesting if it did. But okay. And then, of course, you have chromosomal genetic mechanisms, and that's the case in mammals, where males and females differ in one or more genes or whole chromosomes. Of course, in mammals, the Y chromosome is known to act as a dominant male determinant. Doesn't matter how many X chromosomes you have. If you have a Y chromosome, then you're going to develop as a male. If you lack a Y, you'll develop as a female. So genetic sex is established at fertilization with the inheritance of an X or Y chromosome from the father. However, it's not the whole Y chromosome. It's this single gene that Veronica mentioned, a gene called SRY, that's located on the short arm of the Y chromosome, that is normally essential to trigger male development. So we could prove all of this by doing a simple transgenic experiment where we could simply add a copy of the SRY gene to chromosomally female mouse embryos, and a significant number of them developed as males instead of females. Predictably, these males were sterile. You can also have the opposite, where you find a mutation in SRY, and these have been found in humans and a couple in mice. We can make them now in mice very readily. So if you lose SRY, XY individuals develop as females instead of males. So what is SRY? SRY actually is the gene encodes for a transcription factor. It's a particular type of DNA binding domain called an HMG box. I'm not going to go into any of these details, but so it's a sort of transcription factor. It binds DNA, and the idea is it regulates the expression of other genes. Actually, the SRY gene and the protein it encodes is rapidly evolving. So these are cartoons of the human SRY and mouse SRY proteins. They look very different. The only thing they have in common is actually the DNA binding domain. It seems to be that's really all that's critical about this protein for its role in gonadal sex determination. Okay. Skip the rest, yeah. So if we look at gonadal development in the mouse, this would roughly equivalent period in humans would be around about 6 weeks of development. You have, at early stages, the gonad begins to develop. It's at the indifferent stage, genital ridge stage. I don't know whether these are male or female. I have absolutely no idea. They look identical. But a day later, you can see very distinct differences between the testis and the ovary. You have this formation of a distinct blood vessel in the testis, lacking the ovary. And you have formation of so-called testis cords in the testis, which you get no real changes going on in the ovary. The testis cord has germ cells in the middle, surrounded by a layer of Sertoli cells, a thin layer of so-called peritubular myeloid cells, and then in the interstitium, a variety of cell types, including Leydig cells, which are the cells that make testosterone. So we know in the mouse that SRY is expressed very transiently. It begins to be expressed shortly after the gonads begin to develop. It then reaches a peak of expression around 11.5 days, and then it goes off. In fact, it's off before you see any signs of overt differentiation of the testis. So its role is to initiate the process of testis development, not to maintain it in any way. And the idea is that, so if you consider the normal pathway is to make an ovary. If SRY is present and active, then it diverts that pathway to make a testis. The testis then makes several factors that effectively export the male signal to the rest of the embryo. So there are several cell lineages that make up the developing gonads. This is an oversimplified model, but it's still useful. Many of these are bipotential, so they do something different in the ovary compared with the testis. The critical one, for the point of view of sex determination, are the so-called supporting cell precursors. These are somatic cells, not germ cells. You can ignore the germ cells. If they lack SRY, then they make granulosa cells, and you get ovary development. If they have SRY, they make Sertoli cells. As these begin to differentiate, they then influence the other cell lineages in the gonad to follow the male pathway. Okay. Now, so we're focusing down the question then on how you make granulosa cells versus Sertoli cells. This is a sort of, again, probably an oversimplification, but it lists quite a few of the genes that we know are important initially for making gonads, so there are a whole set of genes. You have, in blue, you have the sort of canonical male pathway, which starts with SRY. Then you have a related gene, SOX9, so it's a member of the same family of transcription factors as SRY, but this is actually a highly conserved member of that family. SOX9 then does all the work, and it triggers the expression of a whole variety of genes that are essential for continuing differentiation of Sertoli cells, and also that influence other cell types in the gonad and ultimately in the body. You have genes in green here, which are required for gonadal development generally, but they tend to many of these tend to have a more critical role in making a testis, particularly, for example, SF1, because it's involved in the regulation of SRY, SOX9, and some of these other genes downstream. So partial loss of function mutations can lead to XY female development in, for example, SF1. Fighting that, you have a set of genes which we can call ovary-determining or anti-testis genes, and there seem to be quite a few that are all acting in a sort of redundant fashion at this stage to try and make sure that SOX9 stays at a low level or off completely in the developing ovary. So these are sort of anti-Sertoli cell, anti-testis genes. So SOX9 plays a really central and critical role. We know that high SOX9 levels give Sertoli cell and testis development. Low levels give granulosa cells and ovary development. We know that from a variety of genetic studies, both in humans and in mice, which show that through loss of function experiments that SOX9 is essential for testis development, or for gain of function genetic changes, that SOX9 is sufficient for testis development. So you can get XX male development, no SRY, if SOX9 becomes active at a higher than normal level in the developing gonad. So it follows that SOX9 is likely to be the only critical gene downstream of SRY. If we look at how SOX9 is expressed during normal mouse gonad development, it's actually expressed at a very low level in both the XY and the XX early gonad. This is before SRY has come on, so you have a similar levels. Once SRY comes on, you dramatically upregulate SOX9, and it stays on at a high level in Sertoli cells throughout life. If there's no SRY, these anti-testis genes effectively repress SOX9, and it stays off in granulosa cells in the ovary throughout life. Okay. So we have a little simplified model for how the expression of SOX9 is controlled. We think at early stages, you have a low level of SOX9, partly because of the action of this transcription factor called SF1, which is required very early in gonadal development. If you don't have SRY, then these anti-testis factors turn off SOX9. If you do have SRY, which itself is regulated in part by SF1, then it boosts the levels of SOX9 expression. Once these reach a critical threshold, they then maintain the high levels of expression of SOX9 through autoregulatory loops. So a former postdoc embarked on a study because it's so critical to understand how this gene is regulated, embarked on a study to try and find critical enhancers, so regulatory regions responsible for SOX9 expression in developing gonad. Now, SOX9 has many other roles in development and in the adult, in stem cells and other cell types. So it turns out that it has such an incredibly complex regulatory region. It's spread over 2 megabases of DNA. It's enormous. So this was an ambitious project from former postdoc Ryo Sekido. He started off with a large fragment of DNA, a back clone. This is the coding region. So it had quite a lot of 5' region and some 3' region. And he made transgenic, he hooked in a reporter gene, beta-galactosidase like Z reporter gene into this. And this gave expression in transgenic animals in several places, all places where SOX9 is normally active. But it gave very nice expression in the developing testis, weak expression in the ovary, if anything. He then cut down this very long piece of DNA and ended up with a particular enhancer, which he called testis-specific enhancer of SOX9, which was about 3.2 kilobases. And this gave very uniquely testis-specific expression, so very robustly. He went a little bit further. He found a core element there, which he called Tesco. I think he wanted sponsorship, but we never got it. Which very, very nicely drives expression, in this case, of a fluorescent reporter gene. Okay. Having such a small element means you can study it in a lot more detail, and we have a lot of information about this particular enhancer element, and this is, sort of, if you like, a simplified model for what's going on, or with this enhancer, but perhaps others too, as you'll see. So the idea is at early stages, you have SF1 bound to this enhancer. It gives you a little bit of expression of SOX9 protein here. At the same time, in the female, you do have actually equal levels of expression of some of these anti-testis genes, in particular the Wnt signaling pathway and the transcription factor that it relies on, beta-catenin, are expressed at very similar levels in both XY and XX developing gonads. And we can show that actually SF1 is bound to its target sequence in this enhancer Tesco, these sequences, and we think is responsible for helping to drive the expression of SOX9 at these early stages. A little bit later, SOI comes on in XYs. This interacts with SF1 and boosts the expression of SOX9. At the same time, in the developing ovary, well, in both sexes, you probably have a slight build-up to even more beta-catenin, etc., but it's not doing anything. And we know at this stage, we can also show that SOI is found bound to its target sequences in this element. If you look a little bit later again, now in the developing ovary, the beta-catenin or whatever these factors are now active, and they seem to work to block expression of SOX9, perhaps through interfering with the activity of SF1. It's known, other experiments, that not only is SOI expressed for just a brief period of time, it's very time sensitive. So if you delay expression of SOI by about 6 hours, it's then too late to work. And the idea is that if you wait 6 hours, you have too much, for example, beta-catenin bound here, and it keeps the gene off. So you have this narrow window of time during which it can work. If you look a bit later, now you've got SOI is no longer needed because you have high levels of SOX9 that then bind the enhancer, interacting with SF1 to give you really high levels of expression. Okay. Let's skip that. So this element, TES, and it's called Tesco, seemed able to drive expression of reporters in developing postnatal testis within Sertoli cells. It seemed able to bind and integrate many of the known positive and negative regulatory inputs into SOX9 expression. And it seemed to be involved in maintenance as perhaps well as initiation, at least from what we could gather from these experiments. But is it essential for expression of SOX9 in the gonad? No mutations have been described in the literature in the human equivalent in cases of sex reversal. So that might suggest that maybe it's not so important in humans. But the obvious question is, what happens if we delete this enhancer from the SOX9 locus in mice? Will it be the same as deleting SOX9, the whole gene, in the early gonad, and will it lead to XY female development? So this is where we get to the genome editing part of the talk. We, of course, used CRISPR-Cas9 genome editing to delete the TES elements of the larger enhancer. Collaborator Peter Koopman did this with Tesco, the smaller element, but we got essentially the same results, which are that you get no sex reversal when you delete either TES or Tesco. So it's not essential. However, you do reduce levels of SOX9 to about half their normal levels when you look at about 13.5 days in testes. So it's important, but it's not sufficient. It's not sufficiently important that when you remove it, you get sex reversal. We can do another experiment where we cross our TES mutant allele with a null allele of SOX9 now, so no SOX9 at all anywhere, and then ask what happens. So you predict that you reduce expression levels down to about 25%. In fact, you get about 23%. Now you start to see signs of sex reversal. You get, this is our control. Here you get clear signs of ovotestis development. The green cells are SOX9 expressing cells. The red cells are expressing one of these anti-testis factors, FOXL2, which is a very early marker of granulosa cells. So the threshold for SOX9 in the mouse is something around about 25%. So we can actually measure the levels, and indeed we do have 23% the normal levels. So TES is required for normal levels of SOX9 expression during testis differentiation, but deleting it is not enough to recapitulate the XY female sex reversal seen in homozygous SOX9 null mutants. That means there are likely to be additional enhancers involved. So a new postdoc, Nisan Gonan, working together with some collaborators, particularly Daniel Matuk, went on a search for other enhancers in this 2 megabases of regulatory region. There's no other gene in this region, hence it's referred to as a gene desert. So they used a variety of methods that look for chromatin in a conformation that suggests that it's bound by transcription factors. These are things like DNase1-seq, ATAC-seq, and then ChIP-seq for a chromatin binding factor. Doing this, 35 candidate sequences were found amongst these 2 megabases. That's an enormous number. So this is looking specifically at cells isolated, purified from the developing testes or ovaries in embryonic stages. Bioinformatically, we could look at these in more detail and realize that, well, 16 were much better than the others. So we chose 16 of these, linked them to a reporter gene, and screened for expression in the gonads using transgenic mice. Okay, again, looking when you should have clearly testes rather than ovaries. And we found of these 16, 4 actually gave rise to expression in the gonads. That's enhancer 8, which is a long way upstream, enhancer 13, which is still 580 kilobases or so upstream, enhancer 14, and enhancer 32, which is actually in between TES and the coding region of the gene. So we got 4. Right, what do these look like in the transgenic assays? Well, in enhancer 14 was sort of what we predicted we would get, where you have very strong expression of the reporter in the testes and nothing in the ovary. But we got other weird things. So we got this, where enhancer 8 gave very strong expression in the ovary, not in the testes. We don't really understand that. We got enhancer 32, which gave very weak expression in the testes, not in the ovary. Actually, if you look in adult mice, it's very strong in the testes. So we think this is probably an enhancer that's more required for adult expression of SOX9. Enhancer 13 was by far the most interesting in a way. I mean, this was important, but this was most interesting. In each case, in each different independent transgenic mouse line we got very strong expression in the testes. But in some, we had expression in the ovary as well. So this said, well, perhaps this is an interesting one. Perhaps it's acting early and it's very poised. It's very, it could go either way depending on integration site in that particular transgenic line. The other reason why we got interested in this is because there have been documented cases of sex reversal in humans involving quite substantial deletions or rearrangements in the upstream region of SOX9. And one of these is referred to as XY-SRY, so these are quite big deletions, many kilobases of DNA, gave rise to XY, was noted to give XY females. So enhancer 13 seemed to map here, at least the mouse, the human equivalent of the mouse sequence mapped there. So This is just showing some of the chromatin accessibility data, very strong peak in Sertoli cells, a weaker peak in granulosa cells. It's a quite well-conserved sequence. So you see it in human, monkey, cow, obviously the mouse, not in chicken, but chickens don't have SRY. Chickens are different, so again, this is interesting. Okay. If we map precisely where enhancer 13 is with respect to this XY SRY region in humans, it maps to the distal end, so it's within it, again in a highly conserved region. Okay. So, what would happen if we were to mutate enhancer 13? So if our theory initially was, well, if TES gives about 55% of SOX9 expression at 13.5 days in the testis, we would predict that no other enhancer could give more than 45%. That's being logical, right? So losing that wouldn't give XY female sex reversal either. Therefore, we first tested the effects of deleting enhancer 13 on a homozygous TES mutant background. Right. So that's what we did. And in fact, we found that now 100% of XY animals carrying homozygous mutations of both enhancers developed as females. So we've got complete XY female sex reversal. However, when we just deleted enhancer 13, we got the same result. 100% gave XY females. These were complete sex reversal. They looked just like females. They had ovaries, they're a bit small, but they had normal-looking ovaries. So enhancer 13 seems to be far more critical than the TES enhancer. It's not simply a shadow enhancer. There's a lot of evidence to suggest that often, certainly in developmentally important genes, that you have several enhancers all doing the same thing. So deleting one doesn't necessarily have much of a phenotype. So this is not simply one of those cases. And this, of course, would fit with the human data on these large deletions of XYSR and the failure to see also mutations in the TES homologous region. So, this is a picture of one of our sex-reversed females with a female sibling. I think it took about 2 hours to get this photograph. They're horrible. And you cannot possibly have a normal male there at the same time. Just all hell breaks loose. You cannot do that. Okay. We can, with lots of work, to show that the sex reversal is complete. So if you look at gene expression, you get absence of SOX9, the same level, it looks the same as the normal ovaries. FOXL2, which is a marker of ovary development, you see it quite strongly, etc. So the complete sex reversal, we can measure levels of SOX9 and some of our other marker genes. And the critical result is this, really. So our homozygous enhancer 13 deleted XY gonads have exactly the same very low level of SOX9 at 11.5 days as a normal control XX ovary. So you basically turn these into normal ovaries. We can look at the expression of, so marker of FOXL2, which is an early marker of ovary development. Again, that looks now equivalent to an XX ovary in here. No effect as predicted on upstream genes, SR1 and SF1. So this enhancer 13 is really quite well conserved, not perfectly, but our favorite transcription factor binding sites you can find in each case in each species, suggesting that this is an important enhancer. We can carry out chromatin immunoprecipitation assays to see whether SRY, for example, is bound to enhancer 13 in vivo. And indeed, we do see it bound, and we find more bound to enhancer 13 than to TESCO. We can also look for SOX9. And we again find that SOX9 is bound, but actually in this case, sort of roughly equivalent levels. This is now looking a bit later. This is at 13.5 days. It's equivalent between enhancer 13 and TESCO. So this enhancer is probably also involved in the autoregulation maintenance phase of SOX9 expression. Not just in the mouse, but this is data from our collaborator Francis Poulat, using ChIP-seq on bovine testes, you also see a strong peak of SOX9 binding on the enhancer 13 in the bovine model. What about the three other enhancers? I'm not going to go through all this because it's quite sort of depressing, but so if you remember, enhancer 14 was testis-specific, enhancer 8 was certainly ovary-specific during embryonic stages, and enhancer 32 very weak in the testis. Deleting these, we got no signs of sex reversal or gonadal abnormalities in any case. So these seem to be very redundant enhancers, or they might have late functions. Just a few sort of broader lessons. Only 1 out of 4 putative enhancers identified by these original chromatin accessibility assays actually show enhancer activity. And of those that show enhancer activity, only 1 in 4 of the novel enhancers turn out to be critical. So that's a hit rate of 1 in 16 to find critical enhancers. But we think, generally this is, particularly SOX9, it's a very complex gene in terms of how it's regulated. So, summary and conclusions of this. There are multiple enhancers, gonadal enhancers. There's also enhancers for all the other tissues it's expressed in. There are multiple gonadal enhancers scattered throughout at least 1.5 megabases of the SOX9 upstream region, gene desert. TES is important at 13.5 days, but it's not essential. Enhancer 13, however, seems to act very early, and its deletion by itself leads to XY female development. This suggests that enhancer 13 not only acts prior to TES and other enhancers, but that it has to. And actually, I've presented some of the data, but we've got a lot of data to suggest that this is indeed the case. This, for some reason, enhancer is acting very early, and it has to act early. Like TES, enhancer 13 is also used by SOX9 to autoregulate its expression, and certainly that interaction we now know is conserved in mammals. Other enhancers may act later to maintain SOX9 expression. For example, enhancer 14 and 32. We're trying to test that. That gets complex experimentally. And moreover, we think that enhancer 13 is likely to be responsible for human sex reversal associated with these deletions of this large XYSR region. And actually, work done by Andrew Sinclair in Sydney, in Australia, which is now just accepted in press, shows that actually you can find smaller deletions associated with this region which remove enhancer 13 in XY females, and very interestingly, small duplications of enhancer 13 can lead to XX male development. So just all you need is a little bit more SOX9, and that's sufficient to then give you, just get over a threshold, you kick in these autoregulatory regions, and then go. Now, lots of people to thank, particularly Nitsan, who did most of this work. She also had 2 babies during this, but it's one of them. We worked a lot with Daniel Matuk in Chicago, who did all these chromatin accessibility assays, and then we had the help of Sophie Wood, who helped us make all these various transgenic and mutant mice, and Francis Poulat with the ChIP assays. Now, how much time do I have?
Well, only about 5 or 7 minutes.
Well, I'm not going to get very far then. So I can either stop here or I can just very quickly go through some of the key issues, I think. So, so I was asked to talk also about regulation of human genome editing. You heard some of this from a different approach about this from Richard. So I was part of a study committee put together by the National Academy of Sciences. It was actually a very international committee, so it wasn't just US focused, which had a look at human genome editing, how it might be regulated. Unlike the Nuffield, who took the approach of framing everything with a sort of reproductive framework, it's sort of parents wanting to have children of a particular type. We took a much more clinically focused look at this. Partly because we didn't just cover germline genome editing, but also somatic genome genome editing, so somatic gene therapies. We can have a typical normal set of principles that you might think of as normal for any type of clinical treatment, and these apply perfectly well here. So these sort of overarching principles, they actually helped our committee make progress, and we didn't argue with each other too much. And but I think they're general principles that should be adopted anyway. So these are promoting well-being, due care, transparency, responsible science, respect for persons, fairness, and transnational cooperation, hopefully. So we concluded that any nation considering governance of human genome editing should incorporate those principles and responsibilities that flow from them into their regulatory structures and processes. There are generally three areas that we looked at. There's research, so basic and preclinical research, basically purely laboratory work on cells and tissues, also on human embryos. So you heard a beautiful talk from Kathy, my colleague Kathy Nayakan this morning. So she's working with early human embryos. Now, we can do that in the UK because we have the HFEA as a regulator. We can even create embryos with an appropriate license. You can create embryos for research in the UK. Now, there are many other countries that do not have appropriate regulations to allow, or that actually forbid any research on early human embryos. So if you're thinking about just basic research, in some countries, many countries, there's already a block on doing even just research. And creating embryos for research, there's only 8 countries that actively permit it. Clinical, 2 types of clinical applications. There's somatic or non-heritable interventions in patients to treat or prevent disease, and then germline, heritable interventions to treat or prevent disease. Generally, people think that dealing with somatic gene therapy so genome editing for somatic gene therapy is the same as we've already done for somatic gene therapy. And it has, using genome editing techniques, has lots of advantage over the older methods. But I'm not going to go into that, they're obvious. But because you can do it more readily and easily, and it's ultimately going to be better than traditional gene therapy approaches where you usually introduce a gene on a retroviral vector, for example, viral vector, that can integrate at random, may not be very active, etc. Because these are much more precise and you're actually correcting the defective gene, we're starting to see an explosion in potential applications for somatic gene therapy. There are now something like 20 trials either started or about to start, which is probably more than the old-fashioned methods ever did. One issue that Richard did not really raise, and we looked at in some depth, was this distinction between treating patients for diseases or avoiding having a child with diseases and enhancement. So we can make a definition of enhancement, which is this, so making changes beyond ordinary human capacities or anything outside of a treatment, prevention of disease and disability. This does pose issues. They were sort of not completely ignored in the Nuffield report, but brushed over to some degree. There's significant public concern about fairness, if enhancements are only available to some people. But of course, we already have many types of enhancement that are tolerated or even encouraged. Nutrition, education, cosmetic procedures. And then sometimes it's a bit hard sometimes to distinguish what would be a therapy and what would be an enhancement. However, at the moment, the range of possible uses of approved therapies for enhancement seems rather limited. And I won't go on to the slide, but at the moment, the risk-benefit analysis, if you're looking at this from a clinical point of view, as opposed to reproductive choice, would say you shouldn't take the risk to do an enhancement at the moment. And we talk about enhancement under somatic treatment because you can do enhancement with somatic treatment as well as you could, in theory, with germline treatment. Let's skip all that. This is a whole area that concerns me. I don't mind garage science so much, but it's the biohackers who want to alter their bodies in some way, or alter the bodies of other people in some way, that for me is the big concern. Because they're already doing stuff which is crazy. It's probably going to harm themselves, but if they propagate it, it will harm others too. And Josiah Zayner was one such person who videoed himself at a meeting, injecting himself with a CRISPR construct to mutate the myostatin gene with the aim of promoting muscle growth. I think he regretted doing that. Okay. So in terms of regulations of somatic therapy, there's already well-established means of doing this in most countries, so it's not a great concern. Let's skip all that. Okay. How about potentially heritable genome editing? Now, we've had methods to manipulate genes in mammals since the early 1980s. With each new one, basically as Richard referred to, with each new one the same questions have arisen about the possibility of using these techniques to treat or avoid heritable genetic disorders, make designer babies, practice eugenics. But it has always been possible to say that the methods are too inefficient or unsafe to apply to humans. But two areas of science open up these debates again. So it's not, I mean, obviously the precise and efficient means of altering DNA sequences provided by genome editing methods, especially CRISPR-Cas9, is very important. But also actually the dramatically increased knowledge of human genes, genomes, genetic variation, and causes of genetic disease all contribute to the excitement around this area. Two possible methods. I'm going to skip this because you've already had talks on this. Okay. All the different methods that Kathy referred to and others referred to have been tried in early human embryos, by the way. However, the methods are still not safe enough to use. More research is needed. At least that's most people's opinion. But it seems inevitable, given the rate of change in the science, that it's going to be made to work both very efficiently and probably safely. Interested by the thousands of inherited genetic diseases, it would allow individuals to have genetic-related children without passing on an known risk of genetic disease. Now, Richard mentioned PGD. I'm going to skip that again, just to stress that PGD is often very inefficient and it's sometimes not possible. So I'm often asked, what sort of genes would you focus on for doing germline genome editing? And it's actually very hard to do that. You can say, well, there's common diseases, such as cystic fibrosis, Duchenne, muscular dystrophy, etc. But there are also diseases that are generally very rare, but occur at high frequencies in specific populations, like Tay-Sachs, Huntington's, etc. And there are over 10,000 single gene disorders. So perhaps it's going to depend on who is actually standing in front of a clinician asking to have a healthy genetically related child. A point you can need to think about is not just now, but in the future. So as our ability to treat patients with genetic diseases improves, and that's including conventional methods, drugs, etc., and the new methods of somatic gene therapy, then there are going to be many more patients surviving to reproductive ages. They will not want their children to inherit probably even a single abnormal allele associated with the genetic disease. So we have to think not just now, but also in the future. Now, lots of stuff on regulation, but I suspect I'm going to have to stop very soon. We came up, at the moment, so certainly in the UK, you would require a change in primary legislation of the Human Fertilization Embryo Act to be able to do this. In the US, the only thing stopping people doing this is actually a rider on an appropriations bill, which has to be renewed every year, which stops the FDA even seeing an application to do this. And it would have to go to the FDA. In other countries, they vary from prohibition, including much of Europe, to possible authorization under strict regulation to absolutely no rules at all. And I'll finish just with this statement. I think we're going to see this happening. We need to think carefully how it should be regulated. Regulations that are flexible are much better than those that are restrictive. Caution is needed, but being cautious does not mean prohibition. So I'll stop there. Thank you.